I'm Tadeo.
I'm Aastha,
I'm Trey.
and today we're going to be doing an experiment testing the rates of photosynthesis.
The materials we used for the experimental group were fluorescent bulb, spinach leaves, hole punch, a petri dish, 300 milliliters of a 0.2% bicarbonate solution, a beaker, syringe, tweezers, and distilled water. Our independent variable was the type of light bulb, and the dependent variable was the rate of photosynthesis.
So the constants that we had in our experiment were the temperature of the room, the bicarbonate solution, and the type of leaf that we used.
To start, we punched 10 equal-sized holes out of the spinach leaves using a hole punch, and then we took the syringe, we took the top out of it, or the bottom out of it, and we put the 10 equally cut circular leaf pieces, and we put them inside of the syringe. Then we put the other part of the syringe back in, and then sucked in some of the 0.2% bicarbonate solution into the syringe so that it was full of the solution plus the spinach leaves, and then we squirted out any air that was inside of it so we could continue with the experiment.
Then we created a vacuum, which means that we just took out the excess air out of the chads, the little pieces of leaf. That way we can make room for the water to come into the chads, and so that way they can sink to the bottom of the syringe.
After they sank into the bottom of the solution, we recorded all of our data with multiple trials, and after all of the trials, we finished our lab and we drew a graph of all of our data collected from this lab, and then we put our materials back into the back lab room, and that wrapped up our entire experiment.
Also, each of the trials that we did were 1 minute long.
The photosynthesis equation is H2O plus CO2 yields C6H12O6 plus O2. Water from soil travels through roots to xylem with capillary action. CO2 enters the leaf from the atmosphere via stomata. Chloroplast absorbs the sunlight. All right.
If we use a fluorescent bulb, the photosynthesis rate will increase because the compact fluorescent bulb emits white light more efficiently and more intensely.
The purpose of the vacuum is to remove the OT from the spongy mesophyll layer so water can enter the chad, making it more dense so it sinks to the bottom.
The chads get CO2 from the bicarbonate solution.
The airspace in the spongy mesophyll filled with O2 as a byproduct of photosynthesis.
When we put chads in the bicarbonate solution, some stack on top of each other. The chads at the bottom of the stacks didn't have light exposure, so they couldn't do photosynthesis as efficiently. Our rate of chads floating could have been reduced because of this making one of our errors in our lab that could have changed our data.
The control group and experimental groups were about the same as shown on our graph, but the experimental group had a higher rate of photosynthesis. It took 4 minutes for about 50% of the experimental chads to rise. We altered the environment because we used a fluorescent bulb instead of an incandescent bulb.
Our hypothesis was correct because the rate of photosynthesis increased when we used the compact fluorescent bulb, as you can see on our data table and our graph.
Hey, Ben. Okay, so I want to ask you a few questions about how your experiment went.
Okay.
So, was your experiment successful?
No, we proved our hypothesis wrong.
You proved it wrong. What do you think made it? What do you think made your hypothesis wrong?
I thought that the heat sink would make the discs go through photosynthesis faster, but it made them slower.
Gotcha. So your experimental group was having no heat disk then?
Yes.
Oh, okay, all right. And what made you think that the heat disk would make it go faster?
Because we thought it was going to be too hot if we didn't have one, and they were just not going to be able to do photosynthesis.
So what you found was that the heat disk didn't have the effect that you thought it would.
Right.
Is there anything in your experiment itself that went wrong?
No.
No, nothing in your experiment itself went wrong.
No.
So for our experimental group, what we did is we changed our normal light bulb into a fluorescent light bulb, and we proved our hypothesis right because we thought that having a fluorescent light bulb would make the rate of photosynthesis a little faster, which it did because in 4 trials we had 5 go up with the fluorescent group, but we had 5 go up in 5 trials for the other group.
Got it.
Yeah.
Okay, go.
Go. All right. I literally just read off the paper.
Yeah.
No, it's not.